So at Aura, we're bringing innovation to ocular oncology, a group of rare diseases that has never had a drug approved. Our focus is to make a difference for these patients, and we're developing a first-in-class drug for choroidal melanoma. We're very excited this week because for the first time, Dr. Brian Marr is going to be presenting the 12-month data of our ongoing phase 1b/2 clinical trial. So what is choroidal melanoma, and why is it such an unmet medical need? It's a cancer in the eye, and it's diagnosed by ocular oncologists. And we're very lucky because we have very early diagnosis of these patients. Unfortunately, at the early stage disease, the ocular oncologists are faced with a terrible decision. Either we observe these patients until we have documented growth with the risk of metastatic disease, and we know that this is a life-threatening disease, or do we treat with radioactivity? And radioactivity, in this case, is showcased here, is plaque brachytherapy or proton beam therapy. It is an incredibly difficult and aggressive treatment for these patients. It creates a number of comorbidities, but most importantly, it creates severe vision loss. So there are 11,000 patients diagnosed globally with choroidal melanoma. And if you look at the segmentation, 70% are in the early stage disease where we could really make a difference if there was a targeted therapy that was able to arrest the growth of the tumor while preserving vision. AU011 is designed to revolutionize the treatment in ocular melanoma. It's an office-based procedure, so forget about surgery or radioactivity. Minimally invasive, intravitreal injections today are standard of care, and also laser activation is something that ocular oncologists are well used to. For the patient, the possibility of having early intervention and preserving vision is something critical. So let's look at the mechanism of action of our drug. This is a first-in-class drug, a novel viral-like particle bioconjugate. Basically, what we're developing is a viral capsid, just a protein, that's conjugated with a small molecule. The viral conjugate is administered intravitreally, and it uniquely binds the choroidal melanoma. We know it doesn't bind the lens, the ciliary body, the retina, very specifically binding modified heparan sulfate proteoglycans that are expressed on the tumor cell surface and extracellular matrix. 6 hours later, once the drug is accumulated in the tumor, a laser beam is applied. Infrared light activates the small molecule and it generates singlet oxygen. This is an incredibly powerful cytotoxic that now it's concentrated at the tumor and generates acute necrosis. This is really a double targeting. We have the first way of targeting through the viral capsid, and then the beam of the infrared light is just shining the melanocytic lesion. That's why we have such powerful cytotoxicity while sparing the retina and preserving vision for patients. So this is a very concentrated market. It's a rare disease. There are only 50 specialists in the US and 50 in Europe. We're already working with the key centers of excellence around the world who are extremely motivated to bring this drug to patients. And there's, as I said, no FDA approved and no one else doing anything for these patients but us. Our current phase 1b study is a dose escalation study, classical oncology, 3 patients per cohort. We started with a single ascending dose, as this is a very novel technology. It was extremely well tolerated. We continued with the multiple ascending dose, and we're currently in the expansion cohort, who will hopefully finish enrollment by the end of the year. I cannot tell you about all of the data because Dr. Marr is going to be presenting it on Monday, but just to highlight one patient that's extremely important. These high-risk patients where the tumor is located right at the optic disc, they have no chance of preserving vision with any radioactive treatment. So what we're showing is, in the graph, the documented growth of the tumor prior to treatment with the red line, After treatment, the tumor stopped growing and had a tumor reduction that sustained for 12 months. But the nice thing, if you look at the vision chart, write the tumor on the optic nerve, and the vision is fully preserved. So the rest of the data of the exciting results that we have will be presented on Monday. We welcome everyone to join us. And again, thank you to our investors. Innovation is brought by a collective team of groundbreaking people. Thank you.